Okay, hello everybody. This is the Unit 1 video to accompany the first assessment for Biochemistry 1 in the 2025-26 school year. In this video, I'm going to quickly talk you through the material that was in the first unit, and hopefully this will be a good reference for you. It's probably not the only thing you'll need to look at or think about or talk about or discuss with me and Kathleen, and anyone else, your peers, who might be helpful to you in order to complete the assessment well. But it is certainly one thing that you'll find helpful, I hope. Another advantage is that you can pause me, you can rewind me, you can speed me up, you can slow me down, you can jump around in the video to get just the part that you need. Any of these things are possible. So, with that said, let me jump right in. This is the periodic table. As you learned, the elements are organized in the periodic table based on electrons, essentially, right? And then, of course, the number of protons going up. So from top left, hydrogen, which has just one proton, all the way up through the table, up to the, over 100 in the bottom of the table. This is an increase in the number of protons or atomic number of the element. We also looked at how each element has an atomic mass, which is based on the protons and neutrons added together. And then in this table, the atomic mass being reported is the average atomic mass, which is if you took all of the different versions of this element that exist, which we call different isotopes. So, for example, if you took all the nitrogen on the planet, right? Nitrogen's up top, nitrogen element number 7. Most of it would be N14 with 7 protons and 7 neutrons, but a small amount would be N15 with 7 protons and 8 neutrons. That would average out if you added up all of the nitrogen on the planet. It would average out to about 14.007 on average, because most of it is nitrogen 14 and a very little bit of it is nitrogen 15. So those we call different isotopes. Remember that in general, atoms of different elements will try to reach the same electron configuration as the elements in the rightmost column of this table, because that is a stable configuration. We've also referred to that as a noble configuration because those are called the noble gases. These are ideal numbers of electrons that are more stable than other numbers. And those in reaching those configurations, the elements in the table will share electrons, right? Such as, say, oxygen often will, or nitrogen often will, or carbon almost always will. They will share electrons with other elements in what's called a covalent bond, or they will fully transfer an electron, such as the example we looked at with Na, sodium, all the way on the left, and Cl, chlorine, all the way on the right, where chlorine can pick up sodium's extra electron. That is called ionization, when the electron is not shared but fully transferred from one atom to another. So either we have covalent bonding, a sharing of electrons, or ionization, a transfer of an electron, in order to get the number of electrons in an atom to be stable or noble in its configuration. Uh, Remember also, and here's some of that info again, just like summarized within a given entry. Here's hydrogen, for example. You see the atomic number, the average atomic mass, here labeled the atomic weight, the symbol, the name, and then some additional info on here, which we're not going to worry about too much in our class, but will come back up in future science class contexts. Um, the actual structure of an atom is shown here. We have, in this particular atom, 8 protons, 8 neutrons, 8 electrons. That would make this, that's right, oxygen. Oxygen 16, right? The particular isotope of oxygen that has 8 neutrons to go with the 8 protons. You could potentially also have, say, like oxygen 15, which would be 8 protons and 7 neutrons, for example. So here's oxygen 16. And note that this is a neutral atom of oxygen. It has 8 negatively charged electrons to go with those 8 positively charged protons. And of course, this is 2 electrons away from achieving a noble electron configuration of having 2 in that first energy level, See those here in this first ring, 2 electrons, the little red guys, pinkish red guys. And then the outermost one here has 6, but if this oxygen could just gain 2 more, it would achieve a noble configuration. So keep in mind, one thing oxygen will often do is to make 2 covalent bonds, and that will cover those 2 extra electrons that are needed. They can be shared in by other atoms, as in H2O, where 2 H's show up and share additional electrons to make a molecule. Okay, here's an example of how oxygen could make a covalent bond, right? And it's shown to you in 2 different models here. So in the top left, there are 2 oxygens not yet bonding, and then you see a black arrow, which shows this is how they could come together to bond. And now if you count them up, the outermost electron shell goes from having just 6 electrons in it, right, on the left side, to having 4 that are fully in one or the other, and then 4 more that are in the middle being shared. And so if you have 4 to yourself and 4 that are being shared, you kind of feel like you have 8, and you've achieved that stable configuration for that outermost energy level. Remember on the previous one, we had 2 in the first level, 6 in the second level. And what this does is gets oxygen up to having 8 in that second level, essentially 4 that are always there, 4 that are being shared. Okay. We would write that because each pair of electrons being shared is a bond. We'd write that as a double bond, shown in the model in the top right, and also shown in this ball and stick model that is in the bottom center of the slide. That's the type that we've been playing with in class. So that's the double bond there between those 2 oxygens. Remember, honk, oxygen is the second letter in honk, signifying it makes 2 covalent bonds. This is a non-polar covalent bond because the atoms sharing here are the same. And so they pull equivalently on the electrons involved in the bond, which means neither one of them wins in that pulling. And that means the electrons are shared evenly, and there's no charge difference from one side of the molecule to the other on average. Okay, on the other hand, water, where H's share with the O, because the O has so many protons, 8 of them, it can pull a little more strongly on the electrons in the bond than hydrogen can. And so you end up with a polar covalent bond. Okay, and that polar covalent bond means the electrons are pushed more toward the O than toward the H. What this means is because the electrons are negative, the O becomes partially negative with that extra electron density around it, and the H's become partially positive with that loss of negative electron density. Because of polar covalent bonding in water, in panel B here at the bottom right of the slide, you can see different water molecules will interact with each other through hydrogen bonds, which are these sticky magnetic-like interactions. We called them sticky-wicky. More on that in a minute. That stick together the water molecules one with another into what's called a cohesive network. Okay. Remember our chant, sticky wicky, not so hot, floats when frozen, dissolves a lot. Cohesion is when water sticks to itself. Adhesion when it sticks to something else that has polar covalent bonds, therefore that has those partial charges that make it attractive to water. Surface tension is cohesion at the surface of a water sample, so that it creates a situation where things can float on top of the water simply by virtue of the surface tension network. Say like a water bug that can walk across water, for example, or a leaf sitting on top of water. And then capillary action, where water moves up the water vessels, the xylem inside of a plant in a tree, for example. Part of that is because the water is sticking to the tree, it's sticking to the wood as it moves upward, and that's pulling it upward. And part of it is because the water is sticking to itself and pulling more water up behind it. So capillary action involves both adhesion and cohesion. Adhesion of the water to the inside of the wood, and cohesion as the water pulls more water up behind it by sticking to itself. Kind of cool. Not so hot refers to the high heat capacity of water. Water can hold an immense amount of heat energy. And this is because water being locked together into a hydrogen bonding network, it's very hard to change the amount of vibration in water. We'll look more at that in a second with a model to illustrate it. Another way of saying this is that water has a high specific heat. It takes a lot of heat to raise the temperature of a gram of water by 1 degree Celsius. It takes a lot of heat to do that. Much more heat than it would take to raise 1 gram of air by the same amount. Okay, and the reason for that is that water being locked together in hydrogen bonding networks, it resists movement. My analogy here was like the rugby team that is hard to hit and make it move more. Water floats when frozen. As we saw, it makes this low density crystal structure. We illustrated this when we practiced these properties with the Kool-Aid activity that we did, and I encourage you to look back at that. Water spreads out and makes a low density solid that floats in its liquid. This is exceptional. This is not what most substances will do. And then water is also an ideal polar solvent, meaning it can dissolve anything else that has polar covalent bonds, as long as that thing is able to interact with water. And so that could include, things like glucose or fructose or citric acid or ascorbic acid, as we did in the Kool-Aid, sucrose. It could also potentially include things that are fully ionic, like NaCl, which we practiced using the water models in class. Here are some illustrations. Cohesion, water sticking to other water. Adhesion, water sticking to other water molecules. Water moving up plants. Surface tension supporting insects that can walk on the surface of water. You can see a schematic of how that works. Feel free to pause this slide if it's helpful. Here's a not so hot slide where you can see I've got this really cool little animated molecule that shows you all molecules that I draw are like, they're not moving, right? Because you need an animation to show movement. But if I could show you what they really look like at all times, this is what they look like. They are constantly vibrating, and the amount that they're vibrating is their temperature. That's not how we feel it, but that is the actual definition. If a thing is hot, that means it's moving more. It's very weird to think about this, but I encourage you to think about this. The fancy way of saying this is the temperature is the average kinetic energy. Kinetic energy is the energy of movement. Okay? Heat's not equivalent to temperature. Heat's a different form of energy that you can invest to change the kinetic energy. So think of it as like if I hit you with heat, I can make you move. So if I invest heat energy, then I can actually increase your kinetic energy. I wouldn't say that my hitting you is the movement. Your movement is the movement. But my hitting into you can change your movement. And again, if I hit into the rugby team, which is water, then it's very hard for me to get them to move much faster. So same amount of heat doesn't always result in the same change in temperature. The reason being that if you hit into something that is on its own, you can make it move a lot, meaning you can make its temperature go up a lot. If you hit something that is locked in a hydrogen bonding network with equivalent force, you won't move it nearly as much. So its temperature won't go up nearly as much, even though you've invested the same amount of heat energy. Here's another way of looking at liquid water on the left, and then the low density crystal form on the right. Again, we drew this out with the Kool-Aid activity, but I hope this is helpful for you to see it and go, oh yeah, it really does kind of spread out when it makes its crystal form, and it leaves a lot of space, which makes it a little bit floatier in its own liquid. It kind of traps a little bit of air on a microscopic level, like a very submicroscopic level. And that spreading out, that lower density causes it to float in itself, which makes for happy Arctic ecosystems, for example, as long as we still have ice there, like this. And then finally, the polar solvent properties of water that you can see here, a salt crystal being broken up and dissolved in water. And notice the way that the partially negative O's of the water are interacting with the positive Na+, that's sodium ion. And then the way that the partially positive H's of water are interacting with the negatively charged chloride ion. Okay. Chlorine as an ion is called chloride because it's negative. That's a naming convention. Okay. Finally, it's worth noting chemical reactions are simply a rearrangement of atoms into new molecules. So for example, here's what happens when you burn something. On the left, you've got burning of hydrogen gas. You can see the hydrogens and the oxygens, which are in their separate molecules, are combined when they burn into H2O. Or in the top right, you've got burning of methane, CH4. that's being turned into CO2 and H2O when it burns, right? I picked some interesting equations, like this is one reason why, one way to reduce carbon emissions is to burn hydrogen instead of methane, natural gas. You can see that methane burning produces CO2, whereas burning H2 only makes H2O. So these are chemical equations that show the way those atoms are rearranging to form new molecules. All of cellular life, all of organismic life, even all of ecosystem level life can potentially be viewed through this lens. Though, of course, you're leaving out a lot if you do that. But theoretically, you could look at all systems, even at very large scales, and think about what kinds of reactions, what kinds of transformations are going on, and that could help you gain insight into the system. Then in the bottom right there, you see an opposite reaction where things can also be popped apart. This reaction is the cutting of water into H2 and O2. Okay. Um, the most important equation that we started to look at in our macromolecules lesson is photosynthesis. That is the most fundamental equation to all living systems, where carbon dioxide and water are combined in the presence of light energy to make glucose, that foundational carbohydrate that is the basis for living systems in many ways, um, and oxygen as a byproduct. Okay, there are different ways of showing that. I've given you a different model at the bottom of this slide, which I hope helps you to think about it. I really hope that you have this equation in your memory. This is a really important equation to actually like practice, write it down, try to test yourself, see if you can still write it down later without looking it back up. I really hope you can remember this equation. We will practice it throughout this year and talk about its importance repeatedly. So starting with just like talking about this and showing you that it makes a carbohydrate one of our major macromolecules is an important starting point for our course. We're also learning that to make larger macromolecules, we can take smaller macromolecules and link them together. And so, with the exception of lipids, all macromolecules do this. Carbohydrates can be linked together, amino acids can be linked together, and nucleotides can be linked together. When you link carbohydrates together, individually, they're called monosaccharides, things like glucose or fructose. They have one ring. They're a monosaccharide. And if you link them together by this reaction called dehydration synthesis, where you pull two H's and an O out and make H2O, hence dehydration synthesis, now you're making a larger carbohydrate called, in this case, a disaccharide. Sucrose is an example of a disaccharide. And if you keep linking them, you can make a polysaccharide. Some of you are producing polysaccharides for your Columbian Exchange annotated molecule project. Folks who are making amylopectin, amylose, those are polysaccharides. Okay? If you're not totally comfortable with that yet, don't worry. We will come back to that. We'll keep practicing it in class. Amino acids are also linked together by dehydration synthesis to make proteins, right? Which are also called polypeptides, chains of amino acids. Those terms are almost interchangeable, and the subtle distinction between them need not concern us at this time, though you're welcome to ask me about it or to look it up. And then nucleotides are linked together to make nucleic acids, as you saw on the model in class, which is one way of looking at DNA. DNA is linked together into this big structure called a double helix, and the building blocks of that double helix are nucleotides. They're also linked by dehydration synthesis. And of course, the opposite reaction that can cut apart nucleic acids into nucleotides, or can cut apart polypeptides into amino acids, or can cut apart polysaccharides into monosaccharides, is called cutting with water, or to use the technical term, hydrolysis, or properly pronounced, it's hydrolysis. Hydrolysis is when you add that water back in that was pulled out during dehydration synthesis and cut the bond back apart so that the units that were linked are now decoupled or delinked, unlinked. Okay, so with the exception of lipids, the other three groups of macromolecules make these polymers, chains of multiple subunits called monomers. And again, to match them up, proteins are made of amino acids, carbohydrates are built into polysaccharides out of monosaccharides, and nucleic acids are made out of nucleotides. Okay. Again, here's another way of looking at that. Amino acids linked together to make polypeptides. But what's cool about this slide is now you can actually see, like, why does this matter? Okay, because I could just be telling you this, but check out on the right-hand side, these are the structures cells are made out of. So amino acids are linked into polypeptides. Polypeptides are linked into things like intermediate filaments, which are, for example, like what muscle tissue contains, right? That's like a cord that you'd find in muscle tissue to make it strong. Um, or, for example, nucleotides are linked together into DNA, and that's what makes up your chromosomes, which are passed on when you reproduce to like cells that you're making, or to your offspring. So that's pretty important. Monosaccharides are linked together into polysaccharides such as starch, and then that starch is stored, for example, in like a chloroplast and maybe used back up during the evening when the sun goes down and the plant still needs to be alive, so it breaks down some of its own starch. Okay. Or to loop fats in, fats can be linked into small groups. Again, not polymers. They're not linked into like chains, but they can be linked into these things called triglycerides, which you see they're labeled as a fat molecule. Again, though, don't think of these as monomers and polymers. They're not. They're just a separate thing, but let's include them. Lipids can basically also be built into parts of cells, such as like energy storage in adipose tissue. Okay. We will come back to all of these structures and more when we go into Unit 2, where we look at how cells are made out of these macromolecules that we're learning about. So it should be that you're feeling the progression in the class here. We're learning the chemistry so we can learn what the molecules are like. We're learning the molecules so we can learn what the cells are like. We're learning biological chemistry. That's what we're doing. Um, Here finally is the fifth and most mysterious of the macromolecules that we're looking at in our chart exercise, the phospholipid. You'll have heard a good bit about this in class, but it involves two fat molecules linked to this molecule called a glycerol with a phosphate on the top. And what that means is that the pink part of this molecule is water soluble because of all of its polar covalent bonds. And that greenish part on the bottom right, those two chains hanging down, those two fats, those are not water soluble because those are all non-polar covalent bonds. And so these arrange in order to make a membrane-like structure. And we'll look more at that membrane-like structure to kick off Unit 2. Finally, as I showed you, I encourage you to kind of think about that these molecules can be represented in beautiful ways. I hope you're seeing the beauty in it. You're welcome to check out this article on David S. Goodsell's work. If you haven't already, I showed you this picture in class already, his rendering of the coronavirus that causes COVID-19. Um, the molecular world is, quite remarkable, and I hope this is getting you excited about it. I hope producing your own representations of the molecular world for the Columbian Exchange project has got you excited about a particular molecule and how it could be represented and how you can come to understand it by representing it. So yeah, I really hope that you're off to a good start here in biochemistry, thinking about how learning about the biochemical basis for living systems is going to inform your understanding of these systems as beautiful, interconnected, functioning systems that have been evolving for billions of years in incredible ways. I'll leave this slide up for a minute. You should pause the video to read through it. These are terms you ought to know, and I'll finish off every unit assessment just by kind of listing them out for you. I'm not going to exhaustively go through them and define all of them. They are things we defined in class for the most part, or that I talked about in this video, or that we looked at elsewhere in an assignment. So I think these should mostly be contextually defined for you already. But I encourage you to go through, and for any of these that you're not totally confident about, or even for those that you are, but certainly for those that you aren't, go through and make a list and have actual definitions. You should be able to look these up. You should be able to find all kinds of great resources on them. And yes, of course, stop by and ask me. Come by at Student Support Block. Ask me for help. I don't totally understand the monosaccharide polysaccharide thing. Can you help me? Yes, of course I can. That's the whole idea. This video is not intended for you to learn every single thing perfectly that is in it. It's just another exposure point for you to think more about it, get more information, understand what you do know about and what you need to know about to understand biochemistry better. And I am here as a resource to help with any of that. That said, that's the end of the video, and I hope you enjoyed it. Good luck with the Unit 1 assessment, friends. See you in class.